Meiosis by Saumya, Narang, and Laor. Meiosis is the process of cell division that creates gametes, like sperm and egg cells. There are three parts of the cell cycle that it entails: interphase, meiosis I, and meiosis II. During G1, the cell grows, develops, and divides. Each chromosome has only one chromatid.
During the S phase, DNA synthesis takes place. DNA is copied. For each homolog, its copy sister chromatid is attached at the centromere.
In G2, the cell continues to prepare to divide. It does this by making proteins, storing food, and replicating organelles. There are now 2 chromatids per chromosome.
Meiosis II is slightly less eventful with the absence of crossing over. In prophase II, centrosomes and their centrioles start to move towards poles of cells, and the nuclear envelope breaks down again.
During metaphase I, the spindle apparatus forms and the spindle fibers attach to chromosomes at the centromere. The microtubules that attach to the centromeres are called kinetochore microtubules, and the ones that don't are called non-kinetochore microtubules. The centrosomes have migrated to opposite poles, and the tetrads begin moving towards the metaphase plate and eventually line up. The law of independent assortment applies here and says that the orientation of one pair of chromosomes is independent of the orientation of another pair.
During anaphase I, each duplicated pair of homologous chromosomes will be attached to a microtubule. Pairs of duplicated chromosomes separate and start to move towards opposite sides of the cell. Cells elongate as microtubules push poles of cells further apart. Duplicated homologous chromosomes now are at separate ends of the cell.
During cytokinesis I and telophase I, the nuclear membrane reforms as chromosomes start uncoiling. Cytokinesis occurs along the midline of the cell, where a cleavage furrow forms as it cleaves to separate into 2 cells, each with their own cell membrane. Each nucleus is a haploid, and the DNA in each of the daughter cells is different from the parent cells.
Meiosis II is slightly less eventful with the absence of crossing over. In prophase II, centrosomes and their centrioles start to move towards poles of cells, and the nuclear envelope breaks down again.
During metaphase II, the mitotic spindle starts to form and to attach to chromosomes at the centromere, just like metaphase I. The chromosomes will begin moving towards the midline or equator of the cell, called the metaphase plate. Chromosomes will be lined up individually along the metaphase plate, and the law of independent assortment still applies here. Each sister chromatid will be attached to a microtubule, as before.
During anaphase II, the cell elongates. Sister chromatids of each chromosome separate and start to move, and centromeres split and chromatids are pulled apart. Kinetochore microtubules shorten. Non-kinetochore microtubules lengthen. Each sister chromatid, now separate chromosomes, is now at separate ends of the cells.
Finally, during cytokinesis II and telophase II, the nuclear envelope reforms and the spindle apparatus goes away. Cytokinesis occurs along the midline of the cell, where a cleavage furrow forms as it cleaves to separate into 2 cells, each with their own cell membrane. Each of the 4 cells is unique and different from their parent cells. In spermatogenesis, there is an equal division of the cytoplasm during cytokinesis, resulting in 4 sperm cells. However, in oogenesis, there is an unequal division of cytoplasm that results in 3 polar bodies and 1 egg cell.